Hello, my name is Polina Shpilker and I will be presenting on contrasting RNA-Seq analysis pipelines for a non-model organism, the coral Pocillopora damicornis. Now, you may wonder why we chose to study corals. The answer is simple. Corals are complex. They are composed of not only the animal host, the coral itself, but also an algae symbiont and other bacteria that make up the complete holobiont or meta-organism. Even a small change in the coral's stress response can cause the entire holobiont to bleach and possibly even die. We want to use RNA-Seq analysis to determine the changes in expression as the coral is exposed to environmental stress to identify genes relevant to the coral's ability to recover from bleaching. However, many RNA-Seq pipelines have been developed and tested specifically for human expression research. Some of these pipelines depend on the fact that we have spent years sequencing and annotating the human genome, and we simply cannot say the same thing for our knowledge of the coral genome. For many corals, we have no sequenced genome whatsoever. Therefore, we ran 3 RNA-Seq analysis pipelines on previously published P. damicornis stress response data, as we have both an annotated genome and transcriptome for this species of coral. We aim to characterize the difference in performance of these 3 pipelines such that we can provide insight to future coral researchers on how to analyze their data. The 3 pipelines we analyzed are combinations of 2 RNA-Seq tools and 2 coral reference databases. The 2 RNA-Seq tools we used were STAR and Callisto. STAR is one of the best tools for analyzing human transcriptomic data, but requires a very high-quality reference genome. Callisto, in contrast, is a pseudo-alignment method that can be run using only a reference transcriptome. For references, we use the databases Reef Genomics and NCBI. Reef Genomics has a P. damicornis transcriptome with some functional annotations. NCBI has both a reference genome and a reference transcriptome, as well as some translations to the Reef Genomics naming schema. Counts from all 3 pipelines were passed to DESeq2 to perform the final differential expression analysis. These differentially expressed transcripts were also passed to GoSeq to obtain differentially expressed Go annotation categories between the 3 methods. In our differential expression analysis, we encountered some issues with naming differences between the 3 references. Therefore, we did not consider any novel transcripts returned by STAR, nor any Reef Genomics transcripts with no equivalent name in the NCBI references. Even with these considerations, we saw substantial differences in the differential expression results between the 3 methods. Most notably, both Callisto pipelines returned far more differentially expressed transcripts than STAR. With an adjusted p-value threshold of 0.001, the Callisto pipelines returned 136 and 129 differentially expressed transcripts, while STAR returned less than half of that, 55 transcripts. Of all of these transcripts, only 30 were shared across all 3 methods, as shown in Figure 3a. On the other hand, of the remaining 106 and 99 transcripts, a whopping 57 were shared between the 2 Callisto methods. We theorized that perhaps a p-value threshold of 0.001 was too stringent for STAR, and that the pipeline returned similar results to Callisto with a higher p-value cutoff. When we increased the cutoff to a p-value of 0.01, STAR returned 179 significantly differentially expressed transcripts, which is much closer to the 136 and 129 of the 2 Callisto pipelines. However, most of these new transcripts were completely unique to STAR, suggesting that the threshold stringency was not causing the difference between these 3 pipelines. Despite the low number of overlapping transcripts, we find many transcripts in similar pathways across all 3 methods. This suggests that even though the transcripts are unique, the many differentially expressed transcripts unique to Callisto are biologically sound and should not be dismissed as noise. To further explore this last point, we performed differential Go analysis on the RNA-Seq results of all 3 pipelines. Interestingly, out of the 44 Go annotations differentially expressed in all 3, only 6 were shared between all 3 methods, and 28 were unique to the Callisto pipelines. Even in the annotations that were shared between all 3 methods, Callisto had unique transcripts that were not considered significantly differentially expressed in STAR. To ensure that these transcripts were not noise, we ran BLAST on the human genome to look for the closest human homologs, and for a select number of these transcripts, there were results. In conclusion, we believe that Callisto returns considerably more significantly differentially expressed transcripts than STAR, but these transcripts are biologically sound and significant. Therefore, researchers working on coral RNA-Seq analysis should not be dissuaded that they do not have a reference genome required to run STAR, and can easily get good results using Callisto.